tertiary three-dimensional architecture. So if you put your fists, make one fist with your right and open your left hand, and you put them together and lock the fingers, that's what we're talking about. Take your right, make a fist, open your fingers on your left, put the fist in your palm of your left and lock it. That's the kind of association we're talking about. That those particular three-dimensional, four-dimensional architectures must be observed strictly for an antigen to act primarily and maximally with an antibody. Next slide. Okay, this is just by the way, we don't really need that. We're in 2020. Now, all antibodies share a basic structure. At any one time, this can only come in an MCQ. No one's never going to ask you to draw an immunoglobulin unless they have lost their minds, which is possible in the current COVID scenario. So antibodies are heavy globular plasma proteins or glycoproteins, and the attached glycans are critically important to the structure and function of the antibody. Each antibody is a heterodimer with a molecular weight of approximately 150 kilodaltons. We shall see that in a diagram. Continue. That's what we're looking at. This is crucial to appreciate. At the core, you see a Y-shaped, huge Y-shaped configuration. On either side of the parallel portions of the arms of the Y, you have a reddish little, tubular structure. This particular design was actually propagated because if you take the three-dimensional configurations of antigen antibodies, especially antibody, you'll never know exactly what's really happening. So to simplify this, they divided it into two sections, the Y shape, blue, and the reddish ones, aptly named heavy chain and light chain. Heavy 2, light chains 2. So this is just to actually amplify what you've had before. All biological systems will require structural or functional biomolecules. They normally occur in 4 chains. Similarly here, just like hemoglobin, but even here we have the 4-chain structure. Now, you can see that 3/4 or 2/3 of the blue Y-shaped antibody or structure of the antibody or the heavy chain seems to be dark blue. And the outer portions of the arms are light blue. Similarly, if you see the tubular, smaller short chain, you have a thicker, more dense colored 2/3, which approximate with the lower portion of the arms of the heavy chain, and a lightish area. This is crucial to appreciate because within the variable region is that portion of the antibody that will make contact with your antigen in 3 or 4-dimension configurations. The heavy chains tend to give structural function to immunoglobulin, whilst also giving you some degree of biological activity. Then you'll see on the right side, you'll see a opened up line reading constant region, as opposed to the variable region on the outside of that reddish component, or the reddish. Both these have a significant function based on the amino acid sequencing. Within the constant regions of both the light and the heavy chains, the amino acid sequence is almost stable. It does not vary at all, as opposed to the outer region, which is called a variable region, where the amino acid sequencing differs markedly. So you can see both variable regions of the light and the heavy chain are side by side, whereas the constant regions extend more or less in tandem. The green little lines you see are called disulfide bonds, and they are very, very strong. In any kind of biomolecule where you have disulfide linkages, they bring about strength to a tertiary or quaternary structure. Suffices to say, the outer parts, where they have the outer parts of the Y shape of the variable regions, are the portion they would call FAB, capital F, fragment AB, antibody, where you see the blue chain indenting, like it's got a sharp turn for it to become parallel, is called a hinge joint. And at the bottom of that, we'll see later, at the bottoms of the two lower portions of your immunoglobulin of the heavy chain will be the part that is responsible for biological activity. So in a nutshell, this is a structure delineating function. But at any one time, this cannot be asked to draw it. We will be asked to know the different components in an MCQ, but it will not be as simple as that. Continue. Next. Yes, an antibody is composed of, hold on, wait a second, slowly, go back one slide. An antibody is composed of two identical heavy polypeptide chains, the dark blue ones, and two identical light chains bonded via interchain disulfide linkages. Each chain is composed of structural domains called immunoglobulin domains. That's just for the purposes of description, but not important, and that they contain 7 to 110 amino acids. This is information as opposed to what is not, it's just, by the way, it's something that you want to know, but not to cram. Now, all immunoglobulins are designated alpha, gamma, delta, epsilon, and mu based on the heavy chain structure. So alpha, we have immunoglobulin A, gamma, we have immunoglobulin G, delta, immunoglobulin D for dog, epsilon, immunoglobulin E, and mu, immunoglobulin M. So each heavy chain has two regions, and we mentioned that, one constant and one variable region. Alpha and gamma chains contain approximately 450 amino acids. That's not important. But just remember, each heavy chain has two regions, one constant and one variable. Next. We have two types of light chains. They are called kappa and lambda. All antibodies have one of the two kinds of light chains. I repeat, all antibodies have one of the two kinds of light chains. This particular importance is very important because you'll be looking at it in hematology, where you'll be having certain immunoglobulin, certain neoplastic conditions or cancerous conditions, where large levels of kappa and or lambda light chains are secreted. And it's important in a condition known as multiple myeloma. So this will be reinforced and has a great or grave importance in delineating different types of plasma cell cancers or neoplastic processes. Just as we described in the reddish little tubular structure, a light chain has two successive domains, one constant and one variable domain. Let's continue. Now, this is where we have everything very well delineated, but we can actually now amplify that which we saw in the simple diagram to more constructive things. Now, for all of us who are there, what can you say about this particular structure now that it has more refined portions? Any comment you'd like to make on this structure? Anybody like to make a comment on this particular structure? You've done some basic biochemistry or basic physiology. Anybody like to make a comment on it? Any comments? Remember when Dr. Gontier is teaching, we have one kind of dogma. Say anything stupid, we shall laugh for 3 minutes, but you'll remain educated. To remain silent is to remain perpetually stupid, and that is highly regrettable. Anybody? Any comment you'd like to make on this structure?
The antigen binding sites are found on the variable region of both the heavy and the light chains.
So you can see like a cup-like structure. Both of those heavy chains seem to have like a little seat on the blue and the green parts. Huh? Anything else you can say?
There are less disulfide linkages between the light chain and the heavy chain.
Okay. So does that make some sense? Yes, that's okay. That's a by the way observation that is not important, but from the point of biochemistry, yes, and more so along the so-called stems of the heavy chain. You have a lot of disulfide. This is a highly strong biomolecule. But more importantly, you must understand, if you see the variable region where it is on the heavy chain is in light blue, and on the light chain is light green. Within these areas, we have what is termed as hypervariable regions. I repeat, what? Hypervariable. That means the amino acid sequence in those particular regions tend to be extremely variable. They actually change a lot depending on the different types of antibodies. And these regions are crucial because that allows us to be able to respond to a lot more antigens than we can possibly imagine. Just to give you an idea, and I keep saying it for the last 15 years, every time the body has got to manufacture approximately 1 times 10 to the power of 9 to the power of 12 different types of immunoglobulins to deal with all a similar amount of antigens. So the combinations and permutations of amino acids in those variable regions has got to be extremely large to accommodate that many antigens to which an immunoglobulin of one specificity of one kind must bind to. If you look at the lower portions of the stems of the heavy chain, we have a portion which is not shown called a FC portion, which we'll mention later. It's a portion that's got biological activity and allows this particular three-dimensional or quaternary structure to bind to receptor sites on different cells. And you'll be seeing it in phagocytosis, you'll be seeing it in complement systems. All of this will have an important bearing. Next. Oh, you've gone through that, yes. Now, these are the important points that you must take home with, and these must be highly reinforced. Because at the end of the day, you'll be receiving reports from the laboratory, especially from the so-called special chemistry. They are not actually special chemistry. It's rubbish. It's just that some particular analytes that used to fall under chemistry used immunodiagnostics. So to cover themselves and to save some grace, clinical chemistry decided to call it special chemistry. But alas, the entire group of analytes uses immunodiagnostics. Things like thyroid hormones, things like female sex hormones, things like drug levels, concentrations in the body, things like the immune response to microbes heavily depend on antigen-antibody reactions. And therefore, this particular part is highly crucial and must be taken with the seriousness it demands. So, most of your MCQs will huddle around characteristics of IgG and so on, and their diagnostic utilities. So let's read this slowly because this is very crucial. They make up approximately 80% of the serum antibodies, IgG. And the half-life of 723 days is a range, but it should actually be 21 days normally, but it has a range depending on different people. but the actual half-life between 20 to 21 days. It is a monomer and has 2 epitope binding sites or 2 antigen binding sites. And this is the only class of antibodies that can cross the placenta and enter the fetal circulation. That is crucial to appreciate. Just go back there. Don't go. I want to really emphasize this and where it has a clinical application, because for us to want to learn immunology, even though it is basic, we must know at this stage how important it is to our future studies in clinical work. So an IgG actually has got 4 subtypes, IgG 1, 2, 3, and 4, and only one of them can cross the placental barrier. And that will be your research to do at home. Which of the 4 crosses the placental barrier and which activates the complement system? I repeat, you shall go back and do a quick look at your Google or the internet and come up with 2 answers. 1, to know which subtype of IgG, either 1, 2, 3, or 4, that can cross the placenta. And notice what is there? That's what I forgot was crucial, IgG. I'll come and I'll talk about it just now. Okay. So when it crosses the placenta, it is responsible for the so-called Rh incompatibility. And not only that one, when the IgG crosses the placenta, it provides the newborn with some degree of immunological protection against hostile environments we can either inhale, ingest, or come into contact with an antigen. So those are protective maternal antibodies. Number 2, there are sometimes when you have mothers who are HIV positive and you'd like to know whether or not the child has been infected, one must never do IgG or IgM for HIV antibodies in the child because the mother will have supplied it during the course of the pregnancy. So you must wait at least 2 years before you can do full testing on the young child, or if it is desperate, you use what is termed as PCR, where we look at where they look for the virus within the blood and its viral particles. Next slide. Now, this is where we're given the first function. The utilization of toxins is vital. Tetanus toxoid that you will get is when you have a cut that's dirty or from an instrument that you doubt its sterility, you will actually be given IgG. It's a powerful antitoxin. And the third bullet, IgG3, binds to Fc receptor by phagocytosis. Let me put this in perspective. IgG is an opsonin. I repeat, opsonin, O-P-S-O-N-I-N. It's an opsonin that encourages phagocytosis by binding through its Fc receptor to a macrophage. So that function of opsonization facilitates phagocytosis, in which the antibody now has a receptor on the neutrophil or the macrophage, where it can bind, immobilize the antigen, and subject and allow or facilitate phagocytosis. Next. IgM is the biggest immunoglobulin in the repertoire of immunoglobulins, and it actually constitutes 10 to 13% of the serum antibodies, and it has a half-life of about 10 days. This is on the lower side. It's normally 10 days and not 5, but some people in different cultures, they can have it as low as 5 years. And it is very crucial because it is the first indicator of exposure to a microbe. It is actually involved in the primary immune response governed by immunoglobulin secretion. Crucial to its function, its structure, most of the IgMs are pentamers. We had the first one we had 2 binding sites. It was a monomer, but here we have a pentamer. 5 monomers, each having 2 antigen binding sites, having a total of 10. So 5 monomers, 2 binding sites each, you get 10 epitope or antigen binding sites. Can occur singly, but don't worry about that. Just worry about its pentameric structure. It is the first immunoglobulin class produced in the primary response. Go next, go the next one. Next. Now, the next one. Good. Just like IgG, which was not mentioned, it activates classical pathway. But remember, both IgG and IgM activate the classical pathway by virtue of having 10 binding sites. The complement pathway is much more easily activated by using IgM, the classical pathway. Defense against multiple antigens acts as an obstacle? No. They've confused this slide slightly. But in any child that you want to diagnose an in utero or in pregnant infection, an infection has come from the mother, the child upon birth can have an IgM screening to the various microbes that you will actually find. So a raised IgM in a neonate, in a child of less than 30 days, is a classical indicator of infection of that particular child in the uterus. All right. And IgM actually binds a lot of polysaccharide antigens, which is used in ABO grouping and cross-matching. Good. Next. IgA. This is very, very crucial. Something that must be taken very seriously because we're going to meet it when we talk about clinical immunology, because IgA is protective to the newborn child and is available in large concentrations in breast milk. Now, there are 2 types of IgA. There's one which is actually secreted into the serum and one which is mucosal bound. So it is thought that since we cannot measure the amount of IgA bound in the mucosa, it might be in higher concentrations than IgG, because we said it was about 80%. Now, protection of the mucosa, all of the mucosas, is the direct responsibility of IgA. Right? It is a dimer, has 4 epitope binding sites, okay, but it's found in all secretions and it is very crucial to the defense of the mucosa. And in some people that will present to your clinic when you're practicing, they will give you a history of recurrent diarrhea, vomiting, coughs and colds, pneumonias. You should be able to order immunoglobulin A concentration, because in non-whites, 1 in 600 people has an inherent deficiency of immunoglobulin A. And this is easy diagnosed and supplemented. So without IgA, you're in a real problem because your entire mucosal surfaces are defenseless. Continue. Just continue. We mentioned all of that here. Next. Now, this one, IgD, over the last 15, 20 years, despite me doing immunology for all that time, nobody's very clear as to what it actually does, but it is an intermediate immunoglobulin type found on developing B lymphocytes. It's thought to be an intermediate marker of developing lymphocytes. We are going from IgG all the way to from an immunoglobulin at step 1, going all the way to immunoglobulin G. It's thought to be an intermediary expression on developing B lymphocytes. Other than that, we're not very sure as to what it's really doing, and research is still ongoing, as far as I know. Next. Leave that one, continue. Yes, this is the crucial one. Now that we're in the third world, IgE is actually very, very crucial. It's in the smallest concentration in blood, but it is very, very, very important because it's the one particular immunoglobulin that gives us the ability to diagnose allergy and parasitic diseases. All right, let's continue. Let's see the next slide. Next. Good. Let's go back to the next one. Go back. Just go back. Go back there. You'll be talking about hypersensitivity reactions, maybe towards the latter half of the course. Things like asthma, things like dermatitis, sinusitis, all those kinds of things. Next. Just flash the next slide. No, the one that came before that. No, before this. No. After IgE. Yes, good. No, after IgE. No. Oh, yeah, there. So note one thing, it has an Fc receptor on basophils and mast cells. These are the cells that are very, very important in allergic reactions. And when an immunoglobulin E binds to those basophils and mast cells, they release vasoactive amines and substances that will cause smooth muscle hypercontractility. So when you'll be doing type 1 sensitivity, you might actually brush over it towards the end of the course. We shall mention the role of IgE and how it interacts with basophils and mast cells. All right. Next, we show the diagrams. Next, good. So you can see, no, go back to the diagrams. Go back to the diagrams. You can see IgG, IgE, D, etc. You can actually see the IgH has got 4 binding sites. You can see the IgM has got 10, got a pentameric structure. And in the middle of that, a joining chain, a chain that binds all of them. They can be bound as a pentamer or singly. Now, what is the diagnostic use of our IgG, IgM, and IgE? Several times you'll be receiving in your laboratory an IgG report. An IgG report might be raised. It only tells you that you have been exposed to that particular microbe before. A raised IgG. If you have a raised IgM, it is indicative of an ongoing, current, or very acute infection. I repeat, it's involved in the primary immune response. Therefore, its levels rises very, very quickly in anybody that has been exposed to a particular antigen or microbe that is actually offensive or not right as such. IgM will rise significantly. If you've been exposed to the same microbe previously, you'll have high levels of IgG. When you have both raised concomitantly or at the same time, you have an acute or chronic disease process. I repeat, if you have them both raised concomitantly or at the same time, it implies or it indicates that you have an acute or chronic inflammatory process. Now, in certain situations, you will be unable to use this effectively if you don't have IgM. A rising titer or titer of IgG 4 times the baseline. I repeat, a rising titer or titer of IgG 4 times the baseline is indicative of an acute infection. These are crucial things that you must appreciate when you go to the wards, when you start learning about going through paraclinical training like in 3rd and 4th year. These are things that you need to be able to understand, the diagnostic utility. IgE levels rise immediately and they have nothing to do with IgG or IgM. But when highly raised, they must be interpreted in light of the disease profile in your area. In the third world, we are constantly exposed to parasitic diseases. So our IgG base levels on normal ranges are much higher. So IgG must be strictly interpreted based on what you're seeing clinically and what disease profiles you have in that region. So if you have high levels of people with asthma, expect your IgG levels to be at a higher range to indicate that if you cross this range or this upper limit, you're talking about having hypergammaglobulinemia E. These are take-home messages because these are where MCQs come from. The latter half of what you've done. In the case of generation of antibodies, the word generation actually refers to an in vitro process, a process that occurs outside the body. And to that, I'll give you just 10 little steps that will tell you how to generate an antibody in the laboratory. In vivo or within the system, that is a process what is known as ontogenesis, the generation of antibodies through a normal process. So all I have to do, I'm just finishing one of your initial things I gave to you on day one. I've got 1 or 2 pages left before I can scan them in, but I'll add these 10 so that we can complete this. I will give the mandate to both or 3 or 4 of the various class reps, the mandate to harass me by Thursday to give you scanned in material of my lecture, the first one, which I think the notes are important, and how to generate an antibody in the laboratory. Simply put, I'll give it to you in a couple of sentences. In the previous 3 decades, to get an antibody was an actual nightmare because you had to take the antigen in its purified form, administer it to an animal, so that within 10 to 14 days, the animal would produce an antibody. You had to go to the various large lymph nodes or lymphoid tissue, even the spleen, macerate it, that means pound it into a pulp, and then after that one, look at separating the cells producing that particular antibody, which we call a plasma cell. For us to identify that, we used to take that plasma cell and fuse it to a malignant plasma cell. And the rationale being is that the malignant cell has the propensity to divide, or ability to divide, without any form of control, physiologically or otherwise. At the same time, you need to do that so you can get a lot of cells producing an antibody from the plasma cell that you got from the animal. So what we used to do, fuse the normal plasma cell producing the antibody from the animal, you fuse it with a plasma cell that is malignant, and you get what is termed as a chimera, or a chimera, a cell that has got 2 distinct portions. One, the tendency to propagate indefinitely, and one that normally produces the antibody that you want. You stick these cells in a cell culture, which has got various reagents that will limit the functions of the various cells, such that only cells that have fused have got the ability to produce normal antibody and to propagate indefinitely are retained. Then you elute that, you get your antibody. To get 1 gram of antibody, you needed half a swimming pool of an Olympic size. But now we do it very easily using various other methods called DNA technology, hybridization. So that's what it is in essence. So when you want to generate an antibody, if you're doing some lab work, PhD, you can go by using a lab animal, but it's not really indicated because we don't need that. It's too long and it's very, very tedious. Is that okay? Okay, thank you very much all. Please, class reps, follow me up so that we can get those two. I'm only 4 pages from finishing the first lecture.